So how we can use the natural product database in our drug discovery research? So if we talk about the traditional drug discovery process, it is a long process that takes 12 to 15 or maybe sometimes 20 years to get get one drug to to become marketed and uh, for the use of for the use of human beings. So if we talk about the drug discovery, it starts from the target identification, lead discovery, lead optimization, and preclinical testing to post approval of the government agencies such as FDA, European Medical Agency, or other based on the different country agencies. So and the if you see the cost of the different phases of the drug discovery is huge. So almost to get one drug approved into the market, uh, the companies spend more than a billion, more than billions of dollars to get one drug into the market. So it, as you can see here, you have to invest your time as well as your money if you start from the scratch. So now we have a lot of databases, we have lot of natural products which have proven biological activity, with the proven biological activity and lot of information of the structure, so that you we can utilize those information and we can use those natural product structure to remodify it or to bioengineerical we can bioengineer them for better activity. So we can save a lot of money and we can save a lot of time, and that's how we can make a good drug molecule, uh, good drug molecule. So in so how we can use this database for the virtual screening workflow, and what is virtual screening workflow? So in natural product drug discovery, if you have large data set, virtual screening is nothing but you are filtering the millions of compounds into just digit compound or three digit compounds. So suppose if you have like millions of compound, as I just shown like in my previous slide, zinc database have millions of compounds. So if you have like millions of compounds, you cannot dock all the compounds into the protein structure, and you cannot see the docking scores for all the compounds, and then you cannot decide, oh, these are the top ranking compounds, so I'm going to select these 100 top 100 compounds for the in vitro activities. So that's going to take you a lot of time. So we cannot do that. So for that, we use virtual screening protocol that saves a lot of our time, lot of money, and lot of the cost, as a lot money and cost as well. uh, so in for that, for the virtual screening, what we need to have, we have to have one protein structure, we have to have some ligands that we can use, and then we can use those ligands and protein, we can do like a structure based designing or ligand based designing. I'm going to show you the steps of the virtual screening. But suppose if you have a target structure which is protein or co-crystallized ligand, means any drug molecule bind with the the particular site in the protein. So we can use those structures, and we can prepare a pharmacophore. We can do the pharmacophore screening. We can take, like, we can do enrichment as analysis, then we can dock the compound, we can do structure based virtual screening, finally we get hit, and then from hit we optimize the leads. So this is just generalized procedure for the virtual screening workflow. So what is the goal of the virtual screening? So as I told you, to find a lead compound that can be optimized to give drug candidate. And what we optimize in the virtual screening? So using chemical synthesis to modify the lead molecule in order to improve its chances to being successful drug. Suppose we have like natural products which are less active or very or inactive against some of the a particular compound. Say for example, we don't want any antagonist, or we don't want any agonist. So what we do in that case, for example, folic acid is a primary dietary source for the human beings, and we are consuming folic acid every day in our dietary products. But folic acid as such do not show any agonist or antagonist effect. But in cancer, in most of the cancer, this folate receptor overexpresses, so we can utilize those folate receptor, and we can we can use this folic acid to deliver drug attached to it. And once that folic acid will be engulfed by the receptor, we can also deliver the drug with that folic acid into the uh, for example cancer cells, right? So suppose we have some natural compounds that are not showing any antagonistic or agonistic effect, so can we utilize them as a ligand just to bind to the receptor which are overexpressing any special kind of disease? So we can also use those kind of natural products. And then we can do high throughput screening that allows millions of compounds. As I told you, we can screen millions of compounds, but the very small portion are available. For example, zinc database, which is the largest data set of the compound, and it is having just like 3 to 4 lakh compounds so far. So we can just use those a small portion of the molecule to design a novel, better drug like molecule. Large scale screening is expensive. So if we want to do large screening, suppose you have 3 4 database and you want to screen them, so you need a good computational system. You will have to have like GPU, or you will have to have a supercomputing facility to calculate the energetics of the docking studies. So this is kind of expensive, but in comparison to overall cost of the molecule, it is very very less. Not all targets are suitable for high throughput screening. Suppose we have like, if we don't know about any target, and if we do not have any protein structure for the compound, and if we do not, or if we do not have any inhibitor molecule for any disease, so we are limited for those kind of diseases or ligands. So we do not have much if we do not have much information about the protein or the inhibitor, so we cannot do the high throughput virtual screen against those protein or ligands. So virtual screening refers to a range of in silico technique used to search large compound databases to select a smaller number of biological testing. As I told you, virtual screening can be used to select compounds for screening from in-house database, or you can also prepare your own your own database, like in-house database. Suppose you have like if you are interested in what you say any piperidine or any quinoline nucleus, so you can compile all the quinoline nucleus all together, and you can generate your own database. And then that would be your in-house database, and then you can screen that in-house database against different proteins to get information whether your quinoline compounds are active against those different proteins or not. So you can do that. That is also possible. Choose compounds for purchase from external supplies. As I showed you, that specs database, zinc database, and there are several database. Those who if you get good hit molecules from them. Suppose you have like 4 zinc compounds, and those are showing good in vitro activity against any of the protein. So you can directly purchase those compounds from that zinc database source, and you can immediately screen then those compounds. Suppose you are getting good AC inhibitory, or you are getting good anti-cancer activity, so you can just order those 4 compounds, and that's going to cost you around 20-30,000, and that will give you idea whether your the lead hit compounds are can be further optimized to get a good lead or not. So that's how we proceed with the this the discovery process. This decide which compounds to be synthesized next. If your compounds are showing good activity, so for example 4 compounds, you have selected 4 compounds, and 2 compounds are showing good inhibitory anti-cancer activity, and two of them are not showing any anti-cancer activity. So you can move ahead with those two compounds, and you can play around the those two compounds. You can optimize the certain groups. You can replace the different pharmacophoric features of the existing compounds, and you can make more uh, 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 more active compounds from the those two compounds. So this gives some better idea. The technique applied depends on the amount of information available about the particular disease target. So yeah, these are some of the general aspects of the virtual screening. So this is the real workflow for the virtual screening. uh, so suppose if you have 3D structure of the target for virtual screening, you need two things. Either you have like the chemical scaffold, or you have the protein structure. uh, I'm sorry, if you have a chemical structure or if you have protein structure. So these two things are required for the virtual screening. Suppose you have 3D structure of the target. If the 3D structure of the target is unknown, right? So you can use this ligand based method. You can go ahead with this ligand based method. And you can select some active compounds, and you can select actives and non-actives, and you can develop one model from this. If you have some active compounds, so you can go for the similarity search or pharmacophore mapping. If you do not have any and if you do not have the protein structure, you can also go with the active and inactive knowns, which is inactives that we call decoys. So you can take some actives and non-actives, and you can make your own machine learning model, and you can screen based on that model. You can screen large data set, and that will give you the good hit molecules. So, and if you have known the protein structure available, so you can do structure based method, and you can do protein ligand docking. So let's start with the similarity searching. So similarity search gives you idea. Suppose these are three things. If I'll ask you which two are most similar, your answer would be orange and basketball, because they are almost circular in shape. They are oranges in color, and they looks almost similar in shape. So I would say, and everybody will say, will agree with this, that these two, orange and basketball, look similar. But they look similar, but they are not exactly similar. uh, yeah, and this looks entirely different. So that's how we get some idea from the this structural similarity search. So what is the rationale of similarity search? The similar property principles states that the structure similar molecules tend to have similar properties, as I told you. uh, Suppose this is the example from the example of the morphine, codeine, and heroin obtained from the papaver somniferum. So these are three compounds that we get from. Though these compounds exactly look similar, like they have like basic nucleus, same basic nucleus, but they have certain different arrangement, functional group, different functional group arrangements, and that's these functional group makes them different from one another. So on the basis of this, we can say no, this will have different property than this will have different property than this. So this, but yeah, but this will seeing the structure, you can say, yeah, if it is showing any activity, a particular activity, it will also show, but we are not sure whether it is going to show that better activity than this or better activity than this. But because they belong to same class, they will be they will be have same they will have some inhibitory or antagonistic or agonistic effect on that particular target. So, basis of medicinal chemistry efforts and all ligand-based virtual screening methods, activity clips, despite the existence of activity clips. What is activity clip? Activity clip are generally defined as a pairs or groups of structurally similar compounds that are active against the same target but have large differences in potency. As I told you, they have different arrangements, and that's what these different arrangement makes them, because of these different groups, their activity will be different. There is no single measure of similarity, and you cannot. There is no. That's true for that's true to the some extent. That is no. There is no single measure for the similarity. You cannot say these are looking similar, so that's how their activity will be similar. Three component of similarity measure. So in there are three components: molecular descriptor, similarity coefficient, and weighting function. These are the three the three descriptors that we use in the similarity search. The first one is a molecular descriptor, and it contains numerical values assigned to the structures, such as physicochemical properties, 2D and 3D properties. Physicochemical properties include molecular weight, log P, and several other the parameters, such as PKI, KI values, different this hydrogen bond donor, acceptor, and so on. And 2D properties, basically the fingerprints, maximum common substructures. 3D property, fingerprints, molecular fields, basically the three-dimensional arrangement of the molecular fields, such as electronegative, electropositive fields. Similarity coefficient, a quantitative measure of similarity between the two sets of molecular descriptors. What are the similarity in the molecular descriptors? And also the weighing function to ensure equal contribution from all parts of the measure. So these are the three descriptors that we use in the similarity search procedure. The next is the scaffold hopping that we use for the similarity search. Scaffold hopping is identification of structurally novel compounds by modifying the central core structure of the molecule. So I'm just going to give you one example that will give you more idea about what is the scaffold hopping. This is the structure of indomethacin. And this drug was developed by the Merck as a pharmaceutical giant. And in this, what they did is they just changed this indole nucleus to just phenyl ring, and the new molecule they developed was the lumiracoxib, and this was developed by the Novartis. So see, by changing this nucleus to this, still it has some pharmacological property, but it is different from the indomethacin. So and that's how you can do several modification into the already existing compounds, and you can make that molecule more or less effective. But I mean, of course, we want to make more effective compounds. So this was just trial by the Novartis that resulted in the lumiracoxib. Further, Merck did some the modification in the indomethacin, and they developed rofecoxib, which has this furan ring, and they also made this toricoxib, which has this pyridine dipyridine ring. So by doing this kind of modification, we can and you can see this is the COX-2 inhibitors. They developed these all are the COX-2 inhibitors. This the Searle Pharmaceuticals developed this celecoxib, which has the pyrazole nucleus instead of this indole. And similarly, Merck also developed Sulindate, which has indene nucleus instead of the indole nucleus. And this drug is like LM1685, has this same indole nucleus but have different properties. So that is how we do the scaffold hopping. We can do some structural alteration in the existing compound, and we can create some compound library, and we can screen them whether we are getting good better pharmacological effect from the existing compound, or we are getting less pharmacological response from the modifications. So we can go ahead with those kind of alterations and the responses from the in vitro or in vitro studies or biological assays. So this is what scaffold hopping is. And the second is pharmacophore mapping. Another part, if you do not have any 3D structure of the protein. So pharmacophoric features of the molecules, there are such as the hydrophobic feature, which gives hydrophobic interaction with the protein backbone. If you have some aromatic ring features, that will also that sometimes also give the hydrophobic interaction with the backbone, protein backbone. If you have hydrogen in like acceptor or donor things in the structure, that will give hydrogen bond acceptor and hydrogen bonding interactions. If you have some cyclic cyclic hydrocarbons or long hydrocarbon chains, that we called as a hydrophobic feature. Other than these, we can also look for the hydrogen bond donors, positive negative features that we can calculate with the Fukui function, customized features, inclusion exclusion volume spheres. So this pharmacophoric feature gives us idea what are the basic requirement of this particular structure to become to show any pharmacological response. Suppose you have this structure and you want to get the same response, so your structure will have to have this kind of arrangement, pharmacophoric arrangement in your structure. Then only it is going to give you that kind of response. So these are some of the pharmacological features. So how this pharmacophoric features, pharmacophore virtual screening works. So if you have like different active compounds, you can select some active compound. You can generate the conformers of those compounds, and you can just modify the pharmacophoric model, and then you do validation of that model. And then you take another hand, you take the select actives and inactive decoys, which is nothing but inactive compounds. So you mix some active compounds and inactive compounds, and you generate a validation database, and then you see, then you cross validate whether your model is showing the same response, whether it is showing inactive compounds as an active, or whether it is showing whether or not it is showing inactive as inactive or active as active. So you can cross validate with your the pharmacophoric the data set, and then you prioritize the selected pharmacophore model, and then you perform the search mapping, and then you do virtual screening. Suppose you have once you get the like all the active and inactive results for the decoys and active compounds, then you generate your own data set, and then you do virtual screening for that. And that model gives you the hit compounds based on your the model generated pharmacophoric model generated. And then you select the compound for a screening. From after the screening, you get the some lead hit molecules, and then you go for in vitro screening. The last one for the ligand based method is machine learning method. In machine learning method, what you do is you take some if you do not have any protein structure, so what you do is you take some compounds. Like suppose you have this C1, C2, C3, C4, 5 compounds. So you substructure you do substructural analysis, recursive partitioning, k nearest neighbors, neural networks. So these are all computational processes, and then you uh, uh, take some training set, and you give some active compounds and inactive compounds to the system, and you generate a model, and then you filter all those compounds to that model, and that model give you some ranking of those compounds based on your inputs. So you get like the rankings like C3, C1, C4, C2, C5. So these are the ranking compounds, and once you get, and then you can choose the top hits or top scoring compounds for further in vitro screening. So that's how the generalized machine learning method works. So, and this is actually more useful nowadays, because what people are doing, they are generating their own AI and machine learning based models, and they are comparing their machine learning, and they are mixing this AI learning and AI and machine learning models with the existing virtual screening or the various pharmacological models, and they are getting good response actually. So this is now more fancy nowadays, after this AI artificial intelligence thing into machine learning tools available. So suppose if you have the structure available, protein structure available, you can do structure based method, and you can do like protein ligand docking. So what is protein ligand docking? So if you have any protein which is showing, which I'm showing you here in yellow, and you can take this this stick ball and stick the stick models here, and this is your chemical structure. You can take this structure and you can dock this structure into the protein specific targets, specific site. Suppose this is the boundary for that active site present in the protein, and you want to dock this structure to this particular site. So once this structure docked into the this particular grid or particular active pocket, I would say active pocket, or so you can see here this portion is going to this pocket, this portion is going to this pocket. It means if it is orienting, this structure is oriented into this pocket like this, it will give you pharmacological response. What if it is like in just imagine opposite case. Suppose you have another structure which is almost both the arms similar, this arm and this arm connected to this phenyl ring. So if it is binding like this will give you pharmacological response. And suppose if this arm will go to this pocket and this arm will go to this pocket, this is not going to give you any pharmacological response. So based on the structure and based on protocol, you will have to design such a model, or you will have to design such protocol that can be used to get good binding interactions. So for evaluating the docking programs, we calculate the RMSD values. So for that, suppose we have these two structures. This is very important for the grid generation. Once we have the co-crystal structure docked into the active site, we compare both the already existing co-crystal structure, and this is the redocked ligand structure, and then we calculate the RMSD value. If it is showing RMSD value less than 2, then we go ahead with that that grid generated grid. Otherwise, we again redock the structure and we do the we recalculate the RMSD value until we get RMSD value less than 2. Then that I am gonna show you in the software. Suppose you have the 3 conformations. So suppose this is the ligand, already existing ligand in the active site, and the brown one is the ligand which we redocked into the active site. So if it is oriented, as you can see here, both the structures are almost aligned to each other, then we say, okay, this is good conformation, and this protein or this grid can be used further for the docking studies. And suppose this has like a little bit fluctuation in the alignment. So as you can see here, this part is almost aligned to each other, but this part has a little bit fluctuation and they are not aligned pretty much. So we can say, oh, okay, it is close, but it is not good. But what if it is like almost different, entirely different? So see, as you can see here, the brown part of this chain is here, and the yellow part is here, and this long tail, hydrocarbon tail, and this yellow hydrocarbon tail is here. So this structure is, we will consider this structure as a wrong orientation, and if we are getting the result like this, we are not going to move further with this kind of arrangement. So further, to evaluate the docking program, what we do is we take a known protein ligand complex from the PDB, extract, and then we extract the ligand from that protein. We minimize the conformation of the ligand. Basically, we do lick preparation. Then we dock back that into the protein pocket, and we compare the dock pose with the experimental data, as I told you, and then we compare the docking RMSD value for that for that particular complex. What are the important energetics for the ligand binding? So if we have ligand protein complex, so we basically calculate the ligand receptor binding energy and the free energy of binding. So this energy, this ligand receptor in ligand receptor complex, we consider electrostatic interaction, dispersion, van der Waals forces, hydrophobic interaction, dissolution, conformational changes, ligand that are oriented and translated in interact from the complex, and loss of entropy. So these are some of the energies that we calculated in the ligand protein receptor. And the another form of the energy is a free binding energy that we calculate in the form of delta G, which is we calculate in the MMGB assay. So there are some other molecular mechanism, force field and stroke functions. I'm going to discuss about these. So as we do not have time, I guess, so I'm not going to discuss these things. But in summary, using natural product databases and computational tools, the new drug discovery process can be potentiated. Natural product databases also provides a way to optimize the molecular escape code, further utilizing total synthesis and semi-synthetic protocol to get it should be to get better drug molecule in the class. Natural product database helps in finding information of the compounds present in the crude extract, and then we can do virtual screening as a robust, and we can utilize the virtual screening as a robust technique to screen larger data set to a few if those can be screened in vitro and in vivo. So with this, my presentation part is over. Now I'm gonna move into the uh, uh, software part. Did you get something from this presentation?
Yes, sir, definitely. We have some learn some
Because I'm gonna show you software part, which is just like do you have access to this software? No. Okay. Okay. So I'm going to launch. Okay.
Yes, sir.
Is it visible? Hello.
Yes, sir. It is visible.
Okay. Okay. So I'm gonna close this. I'm gonna close this card. Okay. So this is the software that we call Schrodinger software or Schrodinger suite, and now I'm going to show you right from the beginning. So I'm just gonna show you. I'm just gonna show you today the pharmacophore generation, the HTVS high throughput virtual screening, and then we'll see some the docking results and how to interpret those docking results. If we'll get time, I can show you some MMGB. No, that is not my part. I'm sorry. So yeah, we will try to cover as much as we can, but I'm not sure. So this presentation was it was long. I'm sorry. So I do not have any natural product database with me right now, but I do have that the small molecular database, specs database, and I have the lick file of that specs database. I'm gonna use that the lick file of that specs database. Just consider that as a natural database, so that will give you more idea. There will be just difference of the structures, but there will be you will get some idea from that. So this is the just interface of the the Schrodinger software. You can do a lot of things with this. How many of you are have used this software before?
Who use the Schrodinger software?
If you are aware with this interface, I'm not going to tell you much about it, but if you are not aware, So I'll just try to give you some examples, or I'll just try to explain little just small things about the software. So initially, whenever we open the software, we'll just go to this file, and we go to the change working directory. And if you won't do this, you cannot find your data, because in our lab, there's a huge lab, and like everybody's working on as everybody works on this the software, and they have like lot of their data. They just generate lot of data. So if you won't choose a particular folder, you can lose your data, you can lose all your data. So whenever we start this software, we first choose the directory. So this is my directory, home, dviker. You can just choose based on your the availability of your directory. So this is my data. So anyway, it is already selected, so whatever I'll do, it is going to be in that folder, that particular folder, and nobody have can access to my personal folder. So everything will be protected, because this is copyright thing. So yeah, that's how we do. First we do go to the file, and we do the this change working directory thing. And then from there, you can also draw the structures using 2D sketcher or 3D sketchers. uh, this is going to take time. Okay, so just close this. So you can just draw the structure using 2D sketcher, and you can save the structure from here. You can give some ID, and then you can save the structure. So that's how you generate the data sets. If you do not, if you have already the data, you have the structures, you can import the structures from your previous file. Suppose I have this. Okay, let me go to another folder. Computer like okay. Okay, so this is the structure. So I have already prepared a specs database, but I just wanted to show you the the small ligand preparation thing. So suppose this is your this is any your what happened? Just give me a second. Okay, so I'm going to select these 10 structures. Okay, and I'm importing just these 10 structure into the table, the working table. Five. Okay, now we have okay, now we have these many structures, almost all these structures. Okay, maximum structures, I think. Okay, We imported 10, not 10 structures, we imported 10 files, I'm sorry. So we have almost 900 structures here, as you can see here. And if you want to see each structure one by one, you can do that. Of course, you can do that just by clicking here. Okay, let me close it. Okay, so this is your first entity, as you can see here. This is your first molecule, right? Okay, so similarly, you can just click on the different molecule, different numbers, and it will give you the exact structure of that particular compound. So further, I'm gonna. So suppose this is our, the database. For example, this is considered this is as a natural product database. So it has 900 structures. Right now, what I'm gonna do is I'm gonna take. Give me a second. Okay. Okay, so I'm just gonna select uh, okay, I'm just gonna select these 13 compounds, and I'm going to export these compounds for our further studies. So suppose this is IFS demo. So I'm going to export this into my folder, which is D Viker, and I'm giving this name. And you can save this these structures as per your, the whatever you like in the different format as well. You can save the MDL, mol to smiley files. You can save in any format, but this software support SDF and .mol file. So I'm going to and MAGZ as well. So I'm going to just convert this structure into .magz file. So, and now I'm going to select all, and then I'm gonna remove this all the structures from this table. Okay. Okay, now we do not have any structure in our project table. Again, I'm going to import a new database that we recently generated, which is IFS. Okay, this is, we'll have to change the format. Okay, here we go. So these are the 13 structures. Okay, I consider this as a, database. It is having like a lot of compounds in it. So what we'll do here is, now we will do leak preparation for the, this is, suppose this is raw data. This is just imported from the, the source file, the source website, or the source database, and now we are going to leak prep. So why we are doing this? Because all these structures are raw in nature. They do not have any particular bond geometry. They do not have any 3D structure, proper 3D structure, 3D arrangement. They do not have their the most stable conformation. So just to make sure all the structures are having their minimum energy, the right orientation, right conformation, we do the leak preparation. The leak preparation generates all the possible structures of that particular compound, and it gives you the most one, two, or three, sometimes four, based on the the conformers generated by that generated for that particular compound. It gives you sometimes one or sometimes two structure for that particular, one or two structure for a particular ligand. So just by clicking here, you can generate all the most possible the generate the most 32 per ligand structures. So, and then you will have to give name here. This is IFS. Yeah, so this is up to this, these scripts we keep as such. We cannot do any changes to this, and no dots, no dash, no hyphens are allowed. Only you can do this underscore thing, shift plus that comma, shift plus hyphen. So, and then you can run this. Sorry, again, no space. Okay. Oh, sorry, I forgot to give the name here. So I'm gonna give here project table 13 entries. Okay. So now we are going to generate. We are preparing the ligands for these 13 structures, uh, and whatever is going on, you can just monitor in the. You can just monitor it here. So right now it is showing you the ligand preparation job is running. So once it is done, it will show you the output file, which is ligprep out file, and that file we are going to use for further HTVS or the docking studies or whatever we want. So now it is showing incorporated finished. It means these uh, the ligands are prepared now for these 13 structures. Then we can close this window and we can go to the project table again. And then you can see here the ligand, the prepared ligands. So we got for these 13 ligands, we got almost 13 structures, which is so this program, this for the 13, we got like each one ligand for each of our structures. Okay, so now I'm again going to close this window and uh, I'm going to go to this virtual screening workflow. So before I'll go to this virtual screening workflow, because I'm doing the virtual screening second part, virtual screening, when our uh, we we have like the known protein structure available. So I'm going to do that part of the right hand side part of that workflow. If you remember, we can do if we do not have any protein structure, and right hand side if we have like protein structure available. So before that we need a protein structure, because this is going to be a protein ligand docking. So we need for that we need a protein structure. So I'm going to prepare a protein first. So protein preparation workflow, I'll go to this protein preparation workflow. So here you can see you can import the protein. You can import the protein from the website, or you can just download the protein from the PDB ID. I'm going to show you here real quick. uh, RCSB. So this is the protein data bank. Whatever the scientists or the researchers, if they generate any co-crystal ligand, or if they do, if they generate any cryo EM or electron micro cryo EM structure or X-ray structures, they submit all the files to this RCSB protein data bank. Uh, so suppose if I'm interested in like EGFR inhibitor. So if I do EGFR, I'll get a lot of different IDs for that particular EGFR, epidermal growth factor receptor. But if you remember, if you have any, if you are interested in a particular ID. So suppose like I'm interested in 2ITY. So I'll just type that 2ITY here, because I know I already work on this PDB, so I know this. So this will show you crystal structure of EGFR kinase domain in complex with IRESA. So this is a IRESA is a drug, anti-cancer drug, and this is already existing along with this protein and co-crystallized form. So we can use this co-crystallized ligand. So if we have like structures available like the co-crystal structure, this is called co-crystal structure. If any drug is present in the protein pocket or along with the protein, that we call co-crystal structure. So we can just use that structure for further HTVS or docking studies. So what you can do here is you can just download the file in the PDB format, or you can do like you can download in different formats, whatever your software supports, whatever format that your software is going to support. But this Schrodinger supports this PDB format, so we can download it here, or we can just go to this get PDB ID, and we can just type 2ITY here, and we can just download it, and it is gonna appear on your screen workspace. This is we call workspace. So see, this now you can see this protein here. This is just raw protein. This is not the prepared protein. So we are going to prepare this protein first, and then we will use that protein for our further HTBS and docking studies. So suppose we have this the protein structure. So what I'm gonna do is next is I'm gonna prepare protein. So the question arises here: why we are preparing protein? What is why we are doing this protein preparation things? Because if the protein is, the protein structure is the set of amino acid chains. So an amino acid has the property, amino acids can the protein chains can fold together. The amino acid can interact with each other, forming the van der Waals bonds, hydrogen bonds, or different kinds of things. So we just want to make the protein in its in its original state, so that it can allow our drug molecule to dock inside the the cavities or the pockets available in between the amino acid fragments or amino acid chains. So that's why we are doing the uh, protein preparation, and we also want to minimize the structure, and we also minimize the energy of the protein. So that's how we are doing this. So this is the protein, and you can also review the structure of that particular protein. So suppose some protein have like more than one chain. If protein is big, it will contain like 2 3 chains. If protein is small, it will just have one chain. So for this protein, as you can see here, this protein has just one chain here, right? And in this chain, okay, I'll go to this protein first. So this is protein. The highlighted fragments indicating the protein backbone, protein backbone. If I and if you want to see the ligand bind with that particular protein, you'll have to see the ligand position. If it has just one ligand, it will show you just one ligand here. And along with that, along with the the ligand, there are certain water molecules are also present in our protein active site along with the protein. As you can see here, 1 2 3 4 5. So before you prepare the protein, or before you start protein preparation wizard, if there are there are 4 chains available. Let me show you one example for that. Give me a second. Okay, let me bring one. Okay, I don't know whether this has that chains or not, but this is a different protein, and it has like 3 4 chains. Yeah, as you can see here, this is a bigger protein than the previous one. As you can see here, different chains. As you can see here, this is a big protein. It has almost 1 2 3 and 4 chains, and then we can see here clearly. So
Hello.
I have,
Sir, can you please change your background as a white?
what is that?
Background as a white. Please change your background. Change your background.
Sir, please change your background as a white. Schrodinger background.
If I'll do that, you cannot see anything. You cannot see the gray scales, and you cannot see the other
Okay,
colors.
okay,
That's
okay,
why I'm
okay.
keeping this as a black. It will be very hard, you cannot see the structure of the molecules and the protein. That's why I'm just keeping it as a black. Is it not visible? So see, I have selected this, and as you can see here, it has 1, 2, 3, 4 lot of the ligands available in the chain. And it has, how many chains it has? One chain, B chain, C chain, and D chain. There are 4 chains available in this particular protein. So this is a big protein, and there are certain water molecules are also available along with the protein and ligand structure. So this is a different protein. I just wanted to show you how many ligands are present in that particular uh, particular chain. So as you can see, like there are like 6 or 7 ligands are present. So it depends what what protein you are selecting. What is the one more important thing I would like to share with you. Whenever you select any protein structure, you will have to see the resolution of that particular protein. The higher the resolution, the better will be the results. It means this is showing 3.42 angstrom, which is not very good, I would say. This is okay. But if it has a resolution like 1.2, 1.2, or 2, so that is good. So always go for the highest resolution, structures that will give you more, confined results when you'll do the docking studies with that particular protein. So that is more that is another important thing. So if you want to study about that particular the protein, what are important amino acids in that particular protein, you can just go to this paper and their paper, because they have developed this structure, so they must have uh, mentioned everything for this particular ligand protein interaction. So you can get all those information from that particular paper, but that is not of our interest so right now. So you can do that later on. So again, I'm going to select this protein. Okay, and I'll go to this protein preparation wizard. Okay, this protein preparation wizard. Again, I'm going to select this workspace entry. Okay, and now you can see here. So sometimes we can delete the water molecule, and sometimes we keep the water molecule as such, because some water molecules play important role in interacting that ligand protein interaction. So if we know, if we have information about that particular water molecule, those particular water molecules, we can keep them as such in the active pocket, or we can remove the water molecules from the active site pocket. So right now, I'm just removing all the water molecules from this structure. So because if you leave the water molecules, they may show you sometimes they will show the hydrogen bond interaction with the ligand, and that's how you get the better the docking score. But when you do the in vitro studies, you do not get the good biological activity. So that's how you'll have to make sure that you are removing all the water molecules from the active site, so that will give you more refined result. But if it, again, I'm mentioning you, if it is important for the ligand protein interaction, if suppose two water molecules are important, then you will have to leave those two molecules inside the active pocket of the protein. Just whenever you are removing anything, just you'll have to make sure you are just doing everything properly. So I'm leaving a chain here as such, and I'm leaving the uh, ligand as such in the protein preparation wizard. Then we will I'm not there are a lot of things, but I don't want to discuss this in detail. It's already too late. But you can, yeah, these are nothing but just minimization, step optimization, and pre-process. So where we do the the refinement, some rotatable bonds and heteroatoms. If some missing chains are there, like in between two amino acid, if one amino acid is missing, so it will, this software will try to accommodate that gap, and it will generate the environment, like that missing amino acid residue will be there. So it's a hypothetical environment. So that software does automatically. That's why we prepare the protein, and now we are going to give that name here. Uh, okay, so and now hit on the run button, and again you can see that here. So it is not yeah, no. So now you can just click on this thing, and you can see the process of the ongoing process for that particular protein. What are the parameters involved in that? The pi stacking, uh, different things are there. Disulfide assignment. So you can, if you want, if you are interested, you can go to all the parameters. What is it is doing in first step, what is doing in the next step. You can track all the steps involved in the protein preparation. So once you have this protein, once you it will prepare the protein, you will get uh, the okay, a different ID. Okay, so once you will get okay, let it finish. It's not going to take that much time, and that will give you more idea how does it work. So it's going to take like few more minutes, and it will be done. Okay, now see it is done. Okay, now here you will see it is done, incorporated, finished. So just make sure once it will show this message incorporated and finished, then only you can import that prepared protein from your the directory. Otherwise, until then you are not supposed to open that file. So once you have your protein prepared, now you can see that will also come in your project table directly automatically. So here you can just click on that and you can see. If you'll see carefully, you will see the different. This protein structure is now different from the the raw structure. It will show you all the the protein, the carbon and the heteroatoms, carbon, hydrogen, and the heteroatoms, and you can see there is no water molecule present here in this. The water molecules you can see in the red ball, small red ball kind of structures. So once you have this protein prepared, you can generate the grid. What do you mean by generating the grid around? So as you can see here, this protein, I'll just show you the surface model first. Okay. So I just want to show you. See, okay, just give me a second. Okay, so now you can see here, this is the protein surface field model, and you can see this is the active site pocket of that protein, right? Where you are that drug molecule is binding to this. Okay, so as you can see, this aniline moiety present in this eraser is binding deeper into that particular pocket. It's very deep. Uh, it's deep, and this quinazoline ring is binding to this another pocket. And this, yeah, this morpholine ring is oriented in the solvent. This is we call solvent exposed region because this is outward from the outward from the pocket. So this is we call solvent exposed region. It is available for like for the physiological fluid or physiological medium. So that's how we can predict. That's how we predict the binding mode of the ligand with the protein. So now here you can see. So for this interaction, if anything going into these two pockets, this pocket and this pocket, and something is coming out of the pocket, that will give you. You can imagine if it is if you are getting the similar binding pattern, that molecule maybe give you the same physiological response, but it is just computational calculations. It is not going to like it is not necessary that you will get if you are getting good interaction, you are getting good ligand protein interaction, you will be getting good biological activity. It's not sure. So this is just prediction. It can give you or it cannot give you the biological response. Okay, so now we will take this particular area and we will generate a grid because this protein structure is big. If we will dark our ligand molecule, it can go and bind anywhere in the protein. So just to avoid that, we are selecting this particular area so that next time whenever we will be doing docking, our molecule will go and bind with this particular area only, not like here, anywhere. So we are now we will generate a box around this ligand. So whenever we'll do docking, our the new ligands will go and bind with this particular pocket. So for that, we are going to generate receptor grid. So this is we call receptor grid. So there are certain parameters that you will have to see. So if there is ligand available in the protein, then you will have to pick that ligand. So right now I am just picking that ligand. As you can see here, it has already selected one box around the ligand, and now it is going to show you the and you can reduce the size of this box as well if and it is based on your expertise and your requirements actually. Okay, so yeah, based on this you can reduce the box like that. You can reduce the box, you can increase the size of the box, but 20 angstrom is average size. It will give you most favorable, not favorable, most accurate results, almost accurate results. But if you select, if you have enough expertise, you can just reduce the size so that the compound will go and bind with the. If you are going to do some special experiments, you can change the size, the box grid size. This is called grid size. Right now I'm just keeping 20 angstrom a grid size, which is a standard. So and then if you do not have any ligand available in the protein, so you can select the x y z scaling. You can select the different residues around which you want to generate your grid. You can select the centroid of that particular receptor. But if we have this ligand available, so we are not going to do that. Okay, so we are just going to generate the grid around the ligand. So now I'm going to give this IFS, and I'll just click on the heat. So before that, okay, let it finish. So I'll just skip this step because I have already prepared a grid file for that. So I'll just import that file directly. Let me clear this everything. Select all, table, delete. Okay. Now I'm going to select import. Okay. This is just grid file, not that. Import. Okay, this is that. Okay. Split. Okay, now I'm just doing, I'm accepting out that ligand from the protein, from the standard protein, not from the grid. So I just I can do that with the see. Now we have this different ligand. We have protein, water molecule, and we have protein. So here you can see this one water molecule is very important for the interaction. Though I have prepared the protein, it was just intact with the ligand and protein complex. So now what we'll do is we will just export this structure and we will prepare the ligand and we will redock this ligand to the protein, and we will see whether it is occupying the same space to that of the original or not. So for that, what we'll do is so this is this is the protein prepare structure for that, and I'm just gonna do this ligand oops, bold, and I'm also gonna do this oops, this ligand bold. Okay, so now, and I'm also going to bring that lig prep ifs. Okay. Okay, so this is the ligand preparation file for the extracted structure. So this is one and this is two. I'll also make them bold so that you can understand it in a better way. So this is the these two are the ligand prepared from that extracted structure, and now I'm going to redock them into the okay. Oops, what happened? Okay, let it complete this procedure. The okay, this is completed. Now I'm going to export this molecule again. Export IFS demo boundary. And now we will do leak preparation for this project table selected entries demo 2. Okay. So I'll show you that later on. I'll go to HTBS. Now this is the very important part. This is HTBS, and now you can select your see. You'll have to have two things here: the ligand file and the protein file. So for that, this is asking for the ligand file. So you will have to select your ligand file, ligand prep IFS, for example, and you will have to select output file and filtering criteria where we do we will check all the boxes. And in the preparation, uh, we do not want to prepare the ligand because we have already prepared the ligands. And receptor, you will have to uh, give the grid file, grid IFS, and then you will have to select the zip file for that, and then you will have to hit OK, and then the docking scores. So this is very important. So if you have uh, so this is the set of the different filters basically. HTBS is nothing but the filtering your compounds from large number to smaller numbers. So this is HTBS is a kind of coarse filter where you this is bigger in size. So where you take like 10, suppose for a 1 lakh compounds, and you select here the 50% or 10% criteria, means you don't want 90% unwanted structures or 80% unwanted structures. So from 1 lakh, it will give you, if you are selecting the 10%, means it is going to give you 10,000 structure. And similarly, you can, in the second step, which is SP, standard precision mode, here you can also select the 10% filtration criteria. It means from 10,000 you are again going to get 10% of that. So it is almost giving you 1,000. I guess, yeah. And then from that structure, if you give the 10 percentage criteria, is gonna give you just 100 structure. This is clear cut calculation, and it is based on your experiment, how many structures at the end you want, and how many structures you have in the in your data set. So, and this process, if you are suppose 1 lakh compounds, it's gonna take like 7-8 days for this the screening, or maybe it depends on your the system, your computational computer system. So right now I'm giving just 10 percentage. So once you do this virtual screening workflow, and you will have to give some name here, suppose IFS demo, and you can just hit this run, and you will get the output file uh, like this, that I'm gonna show you. And then you will then you get the output file VWS. Just go to home. Okay. Yeah. So you finally get this VSW1 specs PI3 something like that. And when you will open this, you will get all the data, your HTVS data, as you can see here, HTVS data, SP data, XP data, everything you will get here. And this quick probe data also you get like in Excel format. And uh, uh, that's how you can see the. I'm just going to open any small file, like I'm going to open that XP file from that uh, with that program. Okay, so from this, okay, I'm going to open this XP file. So see, there were like total structures for around 1,97,000 in that large data set, and from there, from that data set, I am just getting 2, 4, 8, 10, 11 structures finally. And if I compare these 11 structures with my, the interaction to the standard structure, as you can see here, I'll just go to ligand protein interaction, and I'll I'll just go to this bar. As you can see here, there were two sites. So in this particular site, so this is just last part. So here you can see, suppose this is your this is the arrangement of the compound based on their docking score and their ranking, as we were talking about. So if you go to this side of this side of the table, you will see the docking score for that. Different parameters you can see. You can see the QP log value. So basically, we consider in the docking this docking studies, we consider Lipinski rule of 5. If our compound is following the Lipinski rule of 5, that we consider as a good molecule, lead-like molecule. And if it is not following that Lipinski rule, we do not consider that as a lead-like molecule. But this Lipinski rule is a kind of, the not any the hard and fast rule that we'll have to consider. Sometimes the Lipinski, let's suppose for example molecular weight. Most of the natural products shows molecular weight greater than like 500, 600. Sometimes big like paclitaxel. It is a big molecular structure compound. And antibiotics, those are all the natural compounds, but having lot, the big molecular, higher molecular weight. So this is just just for the prediction. So always keep in mind that that to consider the Lipinski rule of 5 when you are doing docking studies. So it says your molecular weight has to be below 500. Hydrogen bond donor and acceptor should be below like 5 and 10 respectively, and Q log p value, which is lipophilicity, should be between 3 to 5. So these are some of the parameters that you'll have to consider always, that you'll have to always consider, because if you are not considering the Lipinski rule of 5, you cannot there may be probability that your compound cannot be like a lead like molecule. So you will have to always consider these few things. So if you go to this side, you will see uh, the different parameters. So first, I would like to show you the glide score or the docking score for the compound that I am getting here, the which is my first hit molecule, and it is showing docking score -12, which is it's a very I mean it's better than the standard eraser. And if we see the structure and the binding of that particular compound, we will see how why it is showing better the docking results. So as you can see here, this molecule is also binding in the active site pocket to similar to that of the standard molecule. So let me show you that. Okay, surface area. Now you can see here. Oh, I'm sorry. Okay, now you can see here. See, this is giving me wrong results. See, this is giving me wrong results because it is showing more interaction, but it is not going deep into the pocket that I wanted. If you remember, I have shown you the structure of the eraser molecule, and it was binding inside these two groups. If you remember, it was binding deeper into the pocket, and just one morpholini ring was coming out of the pocket into the solvent exposed region. As let me show you. Let me show you that as well. Yeah. Okay. So this is that structure. If you'll see here, let me make and the ligand. So now you can see the difference. Now you can see the difference. This particular the compound which is binding inside deeper inside the pocket, and the structure that I was seeing here, it was just binding outside of the pocket. So sometimes we get good results, we get because I'm getting good like docking score. And if based on the docking score, if I say, oh, I got a good molecule which is showing better docking score than the standard drug eraser, so that is just nothing. So you'll have to consider whether your compound is binding exactly into the active pocket or not. If it is not binding, you cannot consider that molecule as active molecule. Though there is a possibility maybe it is interacting with some allosteric site present on the protein because of the structural arrangement of the pharmacophoric feature present on that particular molecule. It is interacting with the different amino acids, which is making it more stable, which is making it more which is making more ligand protein complex stable. That's why it is showing the better result, better docking score as compared to the standard drug. So you will always have to consider the binding pattern of the molecule. If you see the second structure, uh, which is also the same structure for that compound, this is just Okay. Yeah. So almost all the structures are binding on the surface of the protein, not inside the that particular cavity. See, here it is binding into the cavity, that particular cavity from the back pocket. See, this another structure, but it is not in that particular pocket. So I'm just showing you different aspects where what are the possibilities, how and like the compound can interact with the protein and it can bind anywhere into that protein. So never trust on the results. Just make sure you are creating a good model. You are preparing a the machine learning model and artificial intelligence machine mixture of that artificial intelligence and machine learning model, and you are comparing your results of the machine learning model to that of the standard high throughput virtual screening model. So if when you will combine both the models, the probability of getting good results will be higher. So this is all about the HTVAs and docking studies. The next part, I think, Dr. Casey is going to cover. Uh, so this is all from my side. That's it. Thank you so much.
Thank you, sir, for
This is
your
all from my
elaborate.
side.
Yes, yes, yes.
Yes, sir.
Thank you so much. Can I leave now?
Sir, just 1 minute, sir. Just 1 minute, sir.
It's already too late. It's 2:44.
Yes, thank you, sir, for the brief talk on molecular docking and virtual screening. Hands-on training. We are thankful for your ISFCP platform. Thank you very much, sir. Thank you, sir.
Thank you.
Thank you, sir.
Thank you.
Thank you.
Thank you so much for giving me the opportunity. Thank you so
Thank
much.
Any participants want to get your view on the lecture and training, you are most welcome. Any participants from ISFCP as well as outside? Yes, ma'am. Diwali wants their point of view. Diwali. Yes.
करते हैं कैसे से चलेगा
Well,